Hello learners, welcome to Biotechnology Lectures and I'm Dr. D. Prabhavati, here to talk on the experimental proof for DNA as a genetic material. Deoxyribonucleic acid is a biomolecule which is present invariably in all the living organisms and it is a main constituent of the chromosome in the cell. We are well aware of the fact right now that DNA is the carrier of genetic information. It passes on the hereditary information from one generation to the other generation. But well before the discovery of DNA and its function, it was a much disputed question on whether it is a protein or the DNA which is a genetic material. Protein was available in large variety of structure and it had complex functions. Scientists by then could recover the protein very easily and they found them to be in large amounts. So that was convincing enough for them to believe that protein is a genetic material. But DNA, with a structure feature containing of only four nucleotides, it was not convincing for them to believe that DNA is a genetic material. Then came the groundbreaking experiment of Griffith in the year 1928, wherein his experimental strain was Streptococcus pneumoniae and his experimental animal was mice. So Griffith chose two strains of Streptococcus pneumoniae. One, a S strain, which was virulent, capsulated, smooth strain that caused pneumonia in mice. This virulence means the ability to cause disease. So S strain, the virulent factor was present in the capsule and the strain appeared smooth on the smooth colonies on the plate. So that is why it was given the name S strain. So the virulence factor was present in the S strain. The R strain, the strain which appeared rough on the media. The colonies appeared rough on the media. And this was non-virulent. And obviously, they were non-capsulated. So they were rough because of the non-capsulated nature. And they did not cause the disease. So these were the two experimental strains that Griffith took up for his experiment. The control organism was injected with a rough strain, that is the non-virulent R strain. So when this strain was injected in the mouse was alive. In the first experiment, Griffith added or injected the heat-killed smooth strain. He treated the strain with high temperature. So this led to the loss or the destruction of the capsule, and this strain, when injected into the mice, allowed the mice to be alive. In the second experiment, Griffith, experimental observation, which brought to the discovery of the transforming principle. So here he mixed up a rough strain, which is live, and a heat-killed smooth strain. So to his surprise, he found that the mice died out of pneumonia. This he could confirm in another experiment wherein he recovered the bacteria, the live bacteria, smooth strain from the dead mouse, and when re-injected into the another mouse, he found that this mouse also died out of pneumonia. So this was a surprising observation made by Griffith. So good surprising observation was that when injected mice with a mixture of heat-killed smooth strain and a live non-virulent R strain bacteria, the mice died and the live S strain bacteria were recovered from their bodies. So this result suggested that a transforming principle. So he said that some factor or principle was transferred from the dead strain to the non-virulent R strain, turning that into a virulent form. So that is why he named this principle as a transforming principle. And to be noted is that the transformation occurred even though The S strain bacteria had been killed by heat. Though he said that there was some transforming principle, Griffith could not conclude on the nature of the transforming principle. It was in the year 1944, well after the Griffith experiment, three scientists, T. Avery, MacLeod, and McCarthy, identified the transforming principle. So here, the bacterial samples which caused pneumonia, they were treated with enzymes that degraded their proteins, lipids, or RNA. The non-virulent S strain still changed into a virulent S strain, even though the protein was destructed using a proteinase, the RNA was destructed using RNase, still there was no change, and the outcome was the experiment that there was transformation occurring. So the last option was a treatment of the bacterial samples with DNase, an enzyme which destructed or which degraded the DNA. So when this was done, and when this S strain with no DNA was mixed with live R strain, it was observed that the R strain does not get transformed. So, when the heat-killed S strain, which was used in the Griffith experiment, was treated with DNase, it was observed that it did not transform the R strain. So, this meant that the transforming principle was indeed DNA. So, MacLeod, Avery, and McCarthy were able to conclude that DNA is a transforming principle identified by Griffith. But the concluding evidence that DNA is a genetic material came by the Blender's experiment, rather the Hershey-Chase experiment. So Hershey and Chase worked with bacteriophages. Bacteriophages are viruses that infect bacteria. So these viruses structurally are made up of a DNA core and a protein core. So they used radioactive isotopes to label the bacteriophages. The fact that they took to the advantage was phosphorus is not present in the protein and sulfur is not present in the DNA. So they used radioactive phosphorus to label DNA and radioactive sulfur to label protein. So when these labeled bacteriophages infected the bacteria, they found that only the labeled phosphorus entered the bacterial cells. However, the labeled protein remained outside. So this concluded that DNA was the material which is transferred into the bacteria by the bacteriophages, and which led to the production of new viral particles within the bacterial cell. So this experiment provided the compelling evidence that DNA was indeed the molecule responsible for inheritance, which finally convinced the scientific community that DNA was a genetic material. Let's have an illustrated explanation for this. So the blending experiment. So in the batch 1, we have the phages grown with radioactive sulfur. So as we were discussing earlier, this radioactive sulfur labeled the proteins. So when the bacteriophages were allowed to infect the bacteria, the protein coat was left outside and the DNA entered. So when the blending was done and centrifuge followed by a centrifugation, the radioactivity was found in the supernatant, that is, the proteins, and the bacterial pellets did not have any radioactivity. In the second batch, the phages were grown with radioactive phosphorus. So in this case, the DNA was radio labeled and followed by the blending, similar to the early experiment, followed by blending and centrifugation. It was found that radioactivity was found in the pellet. So it meant that the information has been carried on by the DNA for all the bacterial cells. To sum up, the three groundbreaking experiments are the experiments conducted by Frederick Griffith in the year 1928, McCarthy, MacLeod, and Avery in the year 1944, and Hershey and Chase experiment in the year 1944. So Griffith proved that some transforming principle was responsible for carrying the genetic information from one cell to the other, whereas the chemical nature of this transforming principle was convincingly proved by McCarthy, wherein they used different enzymes to treat the bacterial samples, the Griffith experimental strains with the lipase, RNase, proteinase, and DNase, and to convincingly prove that DNA was the transforming principle. Because when DNase was added, the transformation was affected. And finally, the most convincing experiment was done by Hershey and Chase with the bacteriophages. The radioactivity was discovered in the DNA which entered into the bacterial cells. So this experiments proved that DNA is a genetic material. Thank you for listening and join with me for more lectures on the basics of molecular biology. Thank you.